Good morning, everyone. I'm Zhou from UCSD. Thank you for being here. And I want to start by thanking all the organizers for the great opportunity to present my work, and also their effort in organizing such amazing seminars. Today, I will share with you a story about native human GABAA receptors, which we have been published at this year at February. I will start by brief introduction about what is GABA receptor and why we care about them. As many of you know, normal brain function rely on a balance between excitation and inhibition. For the excitation is mainly mediated by the glutamate, but for the inhibition is mainly mediated by a very important neurotransmitter called GABA, which mediates the inhibition by binding to the GABA receptors. So that make GABA receptor mediate the major fast inhibitory neurotransmission in the whole central nervous system. Once the dysfunction of a GABA receptor, which will cause the imbalance between the excitation and the inhibition, which will lead to a lot of excitation tonic disease, which will disrupt the brain normal function. The disease including like the epilepsy, the seizures, the sleeping disorders like insomnia, and also mood problem like depression. All these diseases make GABA receptor a very important clinical drug target and are very important for the pharmacological research tools. So then you may start wondering, what is GABA receptor? They are pentameric ligand-gated ion channel. As a pentamer, obviously GABA receptor consists of 5 subunits. The 5 subunits form a ring surrounding ion conductance channel through the membrane. Once the GABA bind, it will increase the chloride conductance, and then which will lead to the downstream inhibition. Each of the subunits consists of 4 transmembrane helix and a very big N-terminal. In the human, there are 19 GABA receptor subunits, as you can see from here, which including 6 alpha subunits, 3 gamma subunits, 3 Zhou, 3 beta, and also together with some other subunits. All these subunits can assemble each other to form mainly heteropentamer, but some of them can form homopentamer. So easily you can do a math like how many possible compositions of human GABA receptor may exist in the brain. But right now, there's still no very clear idea about how much. The first composition of GABA receptor mainly coming from the localization, which determined by the protein level and also the RNA level. As you can see in different brain regions, the subunits of GABA receptor, they are various. For example, in the frontal lobe, you will see alpha 1 to alpha 5, only 5 alpha subunits, but at the same time, you will see 3 beta subunits and 1 gamma. In another way, like in the cerebellum, you will see 3 alpha subunits, like alpha 1, alpha 4, alpha 6. But here you will only see 2 beta subunits and 1 delta. Based on the distribution and also a lot of classical biochemical assays like CoIP, electrophysiology assay, they propose that the relative possible composition of GABA receptor and their percentage. They propose that the most abundant of GABA receptor is alpha 1, beta 2, gamma 2. Then alpha 2, beta gamma, alpha 3, beta gamma take a little bit lower abundance. Together, they also found some alpha 5, alpha 6, even delta containing composition. Recently, a lot of structures of GABA receptor has been solved. Then I will only show the one matching with the proposed composition here, like the alpha 1, beta 2, gamma 2, alpha 2, beta gamma, alpha 3, beta gamma. But here is not really matching because they also contain an alpha 1 subunit. And also here is the alpha 4, beta delta composition. Most of the structure has been solved through the recombinant structure is not matching with the proposed composition. Since 2014, beta 3 homopentamer came out, and then a lot of heteromeric GABA receptor has been solved, including alpha, beta gamma, even some without alpha, only beta gamma, and some is alpha, only alpha beta, beta delta, and even the homopentamer of Zhou. If this other structure is physiologically relevant, we still don't know. What we know about native GABAA receptor is a paper published at 2023. They mainly determined 3 major compositions from mouse, the rodent. But the literature shows that the compositions of GABAA receptor in the human is much more complicated than in the rodents. So what is the exact composition of human GABAA receptors? We try to answer the question by purify the native GABAA receptor from human brain. So the tissue is mainly coming from temporal lobe and frontal lobe, as you can see from the picture, these 2 different brain regions. All the tissue is from the surgical resections, which is from the epilepsy patients. They are drug resistant, so they have to be treated with the surgery to get rid of the epilepsy. All the tissue are pretty fresh frozen. So there is a specific person will pick up the tissue once they're done with the surgery, and then they will specifically fresh frozen in the nitrogen. But because all the patients, they just do the surgery and they are still alive. So only a small amount is available from each of the patients. So we have to pull in tissue together. As you can see, each of the symbol in the 2 different brain regions represent 1 patient. So in our study, we totally pull in tissue from 81 patients. So then I will introduce what kind of tools what we are using for purifying the native GABAA receptor. Our lab developed an antibody specific to target to the alpha-1 subunits several years ago to help the pro-EM data processing. What we found for this antibody is can specific target to alpha-1, but also at the same time, it shows a very high binding affinity. So we modified this antibody with different affinity tags than used for the purification. But why we choose the antibody only target to alpha-1 subunits? The first very big reason is alpha-1 subunits is very high abundance across the different brain regions. And also the second reason is alpha-1 could assemble with multiple subunits. As you can see from here, all the alpha subunits, all the beta subunits, all the gamma subunits, and also the delta. So we expect to get as much as possible compositions from the human brain with this antibody tool. So then with the brain tissue and with antibody tool, I will introduce you guys how we purify the GABAA receptor with the tissue. All the tissue what we get is in a very tiny amount, as you remember. So they are all stored in the Eppendorf tubes. So then we will accumulate it by getting them out from the Eppendorf tube and then putting them together around 20 milligrams. So then we will use the tissue blender and together with the homogenizer to break up the tissue. Then we will use the high pressure to break up the cell. After several rounds of the different kinds of spin, we will finally get the membrane pellet. Then we will use detergent to solubilize the membrane. At the same time, we will add the antibody to target the protein. As our antibody is modified by the affinity tag, what we use is 3 flag tag. So we can use the flag affinity resin to accumulate the complex of the antibody and the receptor. So then we will put it into the nanodisk. The nanodisk will mimic a native lipid environment. Then we will let the sample went through SEC assay to get rid of the excess antibody and also some contaminated protein. Then we will went through the Western blot for the validation and also the mass spec and even further the pro-EM part. So then I will start to share with you some results. I will first start from the SEC part. As you can see, except the very big aggregate peak, we get another several small peaks. So directly we run SBS page gel to figure out, okay, what's inside. From the gel, as you can see, so many different bands existing there, and we can't barely tell what they are. So what we do is we cut each of them separately, and then we send them for mass spec. Then we have a very clearly idea what they are. So the gel is mainly focused on the first 2 peaks. The first peak is not a real peak, it's like a shoulder peak. And we found it contains the GABAA receptor and also some related protein. The second one is some dissociated related proteins, as you can see from this other part of the gel from here. The last two small peaks is excess antibody and the scaffold protein, what we are used for the nanodisk reconstitution. Then I will give you more detail about the bands in the gel. I will start by the GABAA receptor, which is located at around the 55 kD. As you can see from here, we totally get 13 different GABAA receptors, which including all the 6 alpha subunits, all the 3 beta subunits, 3 gamma, and 1 delta. So which is pretty matching with our previous expectation. I'm sorry. All these subunits multiple subunits can assemble with alpha 1, and we are very excited about this result and hope to get. I'm not sure how many possible compositions from the pro-EM part. At the same time, besides the subunits, you can see so many different bands in the gel. So what they are is NL2, SV2B, and SYT1. Besides these 3 related protein, we also use mass spec to tracking the whole purification process, and we found another 3 related protein, which is GABAA, neuroaxin, and Jeffrin. Besides the last 4 of the protein, the first 2 of them, SV2B and SYT1, they are both synaptic vesicle protein, and there is no clear evidence to show they can target GABAA receptor. So this part is a surprise. For the last 4 of them, they all have been reported can directly interact with GABAA receptor to modulate GABAA receptor clustering and localization. So we are also very excited and look forward to the very big complex structure, not only for GABAA receptor and also with the related proteins. So with such amazing protein sample, we collect a very, very big EM data set and processing for months. Super complicated data processing. You may can see it from paper, but I will not talk through from here. I will directly go to the results about some compositions what determined from GABAA receptor. Total is 11. I will show the first 5 here. The first composition is containing 2 alpha 1 subunits, 2 beta 2, and 1 gamma 2. As you can see, the antibody binding in the binding to specifically the alpha 1 subunits shows in the green color. This is the top view. This is side view of the map. Then the second composition, as you can see in this original composition, the beta 3 can replace the beta 2 subunit. And then even the alpha 1 can be replaced by the alpha 2 subunit. Then in the same way, the beta 2 can transfer to beta 1. And even later, the 2 beta position can be replaced by the beta 3 subunit. Obviously, you can see the beta 1, beta 2, beta 3 can easily switch each other. The same for the alpha 1 and alpha 2 in the first 5 compositions. And all these maps has been resolved at around 3 to 3.4 angstrom, which is enough for us to determine the detailed features. So we can really tell between the beta 2, beta 1, but still at some position it's a little bit confusing. So we give a mixture like beta 2 or beta 3. We also give a relative percentage of each the compositions, as you can see at the top line. So obviously, the first one, alpha 1, beta 2, gamma 2 is the most abundant, which is matching with the previously published native rodent GABAA receptor and also the functional papers. So then I will start to introduce another 6 compositions what we get. The first one is a very interesting one, which is a real, heteropentamer, which consists of 5 different subunits. As you can see, it's alpha 1, alpha 2, beta 1, beta 2, and also gamma 2. The second one, you will see the similar composition, which is beta 2 replace beta 1. And then beta 3 can replace beta 2. And also another side of beta-3 of beta-2 has been replaced by beta-3. Even later, we also can see alpha-3 existing in the compositions. Until right now, I have totally introduced you guys around 10 compositions, and they all follow a very classical GABA receptor assemble format, which consists of 2 alpha subunits, 2 beta subunits, and 1 gamma. Our last composition disrupt this rule with containing 3 beta subunits, which contribute a beta-beta interface, which is never existing and also reported in the native rodents, and also the beta-gamma interface. But this composition has been reported previously by a similar composition from recombinant GABA receptor, which is kind of like one validation from our results. So from all these 11 maps I just introduced, you may found the really complicated composition, and you saw the super high heterogeneity of native human GABA receptor. So that's the reason why we care about GABA receptor, because the heterogeneity make a GABA receptor are very rich in the pharmacology. So GABA receptor pharmacology is mainly determined on the different interface which contribute by the subunits. So as you can see, the different subunits contribute different interface, which is specifically for different kind of drugs binding. And I hope our results will provide some insights for the clinical drug design for the human GABA receptor. Then I will introduce 2 very interesting points we found for the data processing of the native GABA receptor from the human. The first one is we found a rare density across the different GABA receptor composition. The first type across 3 total compositions, they all located at the extracellular domain of the alpha-1 and the gamma-2 interface. This interface is pretty popular, which has been reported for a lot of clinical drug binding. One example here is a drug called Valium, which is using for treating the anxiety and seizures. So here is the pocket where it binds. As you can see, the green color is the density map. What we get? They shows in different shapes, which may present a mixture binding at this pocket in different composition. And also, at another interface, we also found the mystery density, but this one is located at the alpha-beta interface, also in different composition. The last one is located as a beta-beta interface, which is shown in the yellow color. This interface has been reported for the histamine binding. For the alpha-beta interface, which never been directly reported for any drug or clinical design specific drug binding. Yeah, so there is no evidence to show the binding at the alpha-beta interface. All the density, what we found at all the interface is very strong. Even we change the threshold of the map, they are still there. So all this mystery density encourage us to move forward. Okay, maybe it deserves for us to investigate what may contribute to them. The first which came to our idea is the endogenous ligand in the human, which has been identified combined to the GABA. So, for example, is the endogenous neurosteroids and the endozapines. But all these endogenous ligands, due to the binding localization and also the binding affinity, which may wash out during the purification, is now matching with the results showed here. So then what really attract our attention is all the anti-epileptic drugs taken by the patients. All the patients are they still taking the medicine just before the surgery. So we summarize all their information and finally found, as you can see shown in the different color of the frame, mainly 6 different types of the medicine, which has been taken by a lot of patients. And we investigate each of them. For example, the first one, lacosamide, among 81 patients, 35 of them are taking it. This mechanism of the lacosamide is by enhancing the sodium channel inactivation, and then is the lamotrigine and the LEWA zonisamide. For the last two types, they all has been proved they can target the GABAA receptor, even with a little bit lower number than the first four types. But we still think the last two types may contribute to the density what we found. But because they can already target GABAA receptor, we are not very interested in this part. What we are thinking is for the first four types, they have been found to target sodium channel, calcium channel, and even SV2A protein. And a lot of patients has been taking this kind of medicine. If they might have any effect on GABAA receptor. So what we are doing is we use the electrophysiology patch clamp to test the activity on GABAA receptor channel. And we test 4 of them. But finally, we found 2 of them, the lamotrigine and LEWA. They can slightly potentiate GABAA receptor in the composition of alpha-1, beta-2, and gamma-2. And putting together, we found that this result is pretty significant. So in this composition, it contains different interface. For these two, alpha beta and alpha gamma is the position where we found the mystery density. So then we are keep pushing us like to move forward to, okay, where these drugs really bind to the GABAA receptor. So then we use another composition is alpha beta to determine where the drug may function. But the result is pretty surprising, and we found previously it's potentiated, but later when we change to alpha beta, it's slightly inhibit the GABAA receptor of alpha-1 beta-2, and the result is also pretty significant. Then we check the compositions. What we found is even if we get rid of gamma-2, we get rid of the alpha beta interface from the first part, but we introduce a beta-beta interface, which also found some mystery density. So we think the beta-beta may contribute the inhibition, and the alpha-gamma may contribute the potentiation. But from here, the conclusion is still not very clear. We still want to figure out where this kind of like drug plays such a kind of like interesting role. So what we do is move to pro-EM. We try to resolve some recombinant GABAA receptor with both the lamotrigine and also try to determine with LEWA. But finally, we only determined the one with lamotrigine binding. And what we found is it directly targets to the alpha gamma extracellular domain interface. And also here is the binding pocket, and you can see the density fit the lamotrigine ligand in a very well way. As you may remember, we're talking about the mechanism of lamotrigine. It targets to the sodium ion channel. Sodium ion channel is involved in the excitation of the neurotransmission. But now we found that lamotrigine also can involve in regulate the inhibition of the neurotransmission because it's targeting to GABAA receptor. So we hope our results will provide some interesting idea about, okay, how lamotrigine works in treating epilepsy, not only through the excitation part, but also through the inhibition part. So then I will tell you the second, but also the last very interesting point is also during the composition determination. What we found is in the composition with 2 alpha-1 subunits, 2 beta and 1 gamma. What we found is extra density, which is located at the transmembrane domain of the gamma subunit. As you can see in the red color, the density, and it shows in the 2 different views. So what may contribute to the density, it reminds us to think about our great idea about the super big complex structure. And then we went through each of the potential candidates for the SV2B and SYT1. As we already discussed, it's a presynaptic protein, and there's no evidence to show they can interact with GABA receptor. For the NR2 and GAR4, there is already published literature to show they can target to GABA receptor and specifically through the gamma transmembrane domain. Then is a neuroaxin, which can directly target to GABA receptor based on the published paper, but it's also only located at the presynaptic site. For the Jeffrin, it's soluble protein and only interact with the intracellular loop of the receptor. So based on the mass spec results, we think NR2 and GAR4 really matching the candidate. So we keep moving on with the model docking. What we do is we use AlphaFold to predict the transmembrane domain and the whole structure of GAR4. And then we fit it into the density. We found it fits directly very well, even we didn't do any further refinement. So we think, okay, it could be NR2 and GAR4. And if you check the interaction from the side view, you will see the GAR4 work like a bridge to mediate the interaction between the NR2 and the gamma subunit of GABA receptor. This part is really matching with the published functional paper. But I want to say for this part is still pretty limited because our resolution is very low. It's just around 4 angstrom. It still needs further validation. So then I will give you a summary about my today's talk. We totally determined 11 maps of GABA receptor, and we found heterogeneity super high. And we found some unique interface and subunits only existing in the human compared to in the rodents. And we also found all the subunits are corresponding to our mass spec results. And also we found some rare density in the maps, and which lead us to investigate two drugs, LUA and lamotrigine. They have some activity on GABA receptor, and finally determine the structure of where the lamotrigine bind to GABA receptor. And then based on the mass spec results and model docking, we suggest the complex structure of the GABA receptor together with NR2 and GAR4, but they still need further validation. So then I will end of my talk by the acknowledgement. I want to thank my supervisor, Ryan, for being so supportive and all the guidance through the project, and also Colleen for the model building. All the patch lab assay in the paper has been done by Jinfeng. And also we want really thank the donors of all the epilepsy patients to make this research happen, and Dr. Liga and Hailey for the tissue carting and frozen, all the data sets is collecting at UCSD cryo facility. Thanks Mar for the nice help. All the mass spec is done at UT Southwestern by Andrew. And also I want to thank my HA postdoc fellowship, and thank you all for the listening and attention.
Thank you very much, Jia. That was really lovely. So for those of you who are new to the seminar, you can type your questions in this question and answer little bubble at the bottom of your screens. There are already some questions in the chat. I would just, if I may, just start off with one question. So if I follow your purification pathway, you pull down GABA receptors from the brain using the antibody. So in that pull down, how many of these receptors do you think are ER receptors, intracellular? How many of them do you think are actually synaptic and extrasynaptic? And do you think that would affect the subunit composition of the receptors?
Okay, that's a great question. To be honest, we can't really tell where it's really coming from. Because we just do the region, we pulling all the tissue together. We can't really to do the cell level, even the presynaptic and synaptic and extrasynaptic. It's super hard to define the thing in that way. But based on our result, I think what we are getting mostly is synaptic GABAA receptor, because they are all gamma-2 containing GABAA receptor. So that's the previous idea from a lot of functional papers. They propose the gamma-2 containing is mostly synaptic GABAA receptor. So yeah, so that's the only way from the conversation we can tell it's synaptic or extrasynaptic. But from the previous, that in opposite way we can't really tell. Yeah.
Okay, so I'll go to the chat now. So the first question is asking, did you use lipids extracted from the cell lysates when reconstituting the complexes into nanodiscs?
Yeah, we use the so the question is if we use the nanodisk for the
so yeah, but which lipids did you use in these nanodiscs?
Oh, the brain polar lipid.
Okay, so it's the lipids extracted from the brain, right? From the
commercial form, the Pauling brain polar lipid.
Okay. Okay. So the next question is, what EC of GABA are you targeting for the EPIC experiment? So 1 micromolar on alpha 1, beta 2, gamma 2, and 300 nanomolar on alpha 1, beta 2 may not be the same EC.
Oh, great catch. To be honest, at the first, we use the same concentration. We all use 1 micromolar, but later when we start to see the inhibition from the alpha base side, we found after at 1 micromolar, the inhibition is not very stable. Until when we increase the concentration of GABA, we can see, okay, it's very stable, and then we can finally conclusion, okay, there is the inhibition. Because also for alpha 1 and beta 2 composition, there is a rundown. So after at 1 micromolar concentration and also rundown together, it's super hard to tell. So we keep testing the concentration, and we finally found at 300 micromolar, it's pretty stable, and it will give us a very clear results. So then, that's the reason why we pick up in the different way.
Okay, thank you. So now we have a comment from David, who did a lot of single channel studies on pentameric ligand-gated ion channels, and this is what he had to say about GABA receptors specifically. Unsurprisingly, when we looked at GABA receptors using single channel recordings, we found them to be too heterogeneous to allow mechanistic studies. The heterogeneity would not have been detectable in whole cell studies. So a lot of work they then did on glycine receptors, which were definitely much better. So that definitely agrees with what we are seeing in terms of subunit promiscuity of GABA receptors, and we have a similar phenomenon with AMPA receptors. Leila, did you want to ask something?
Yes, I did. That was really, really beautiful and really brave work.
Thank you.
I was wondering, and I don't know if this is at all known, but is there a difference in subunit composition between epilepsy patients and non-diseased human beings?
Oh, that's a great question. Yeah, that's a super great question. Yeah, we also get that question from our reviewer during the paper review. Actually, there is no perfect control we can use. Yeah, so what we can normally get is postmortem people, also from tumor, they cut, then you get partial of the tissue. So you can't really compare if the epilepsy patient can, the composition what we get from the epilepsy patient represent the normal composition for GABA receptor. But what I can tell is there's some published literatures, they test at least from mouse, from the rodents, the clinical model of the mouse, and they present kind of like a similar composition, or kind of like same composition with the normal mouse. And also what they are testing with the tumor, the tissue cutting from the tumor diseased person. And then they do some EPIC assays. They found, okay, it's kind of like a very similar, at least for the major composition of GABA receptor. But indeed, the heterogeneity of GABA receptor is super hard to tell, even through the electrophysiology and all the assay. We hope what we present is the mainly compositions in the normal people. Yeah.
Well, I think it's interesting. It's interesting either way, because I would have thought, maybe in epilepsy you would have, you would have maybe a bias towards subtypes that are, or compositions that are less receptive to GABA binding, so you have less inhibition. But it's really fascinating either way.
Yeah, and also epilepsy patients, a lot of them will share kind of like mutations in GABA receptor. And we try to figure out, but that's something we didn't really find. That's very interesting, and but we didn't really get. And normally, how to say, the monthly subunits, what we get is kind of like matching with the normal people. Yeah.
Okay.
Yeah. Thank you.
All right, thank you.
So going back to the GABA concentration, the next question is, how physiologically relevant are the concentrations of GABA used to test for the effects of LTG and LEV drugs?
So the question is, what the GABA concentration for the I just I'm sorry.
So the question is, how physiologically relevant are the concentrations of GABA used to test for the effect? So this slide basically, so the 300 micromolar GABA and 1 micromolar, is this something that GABA receptors would see in the synapse, or do they see different concentrations?
Okay, yeah. I'm sorry. I just thinking about like the drugs and the GABA. The GABA in the physiological concentration is kind of like keep changing. The highest can reaching even millimolar.
Millimolar, right.
But it's pretty stable. It's around micromolar. So EC50 for GABA, for direct GABA receptor, is around like 30 micromolar. So 1 micromolar is indeed under the physiological concentration for most of the testing. But I still want to say 300 micromolar is not very high, because it's when it's kind of like keep changing, it can reach millimolar level. So
okay. So kind of building onto that is a comment that alpha-1 beta-2 gamma-2 receptors are postsynaptic, and thus potentiation may not be seen at physiological concentrations of GABA. So the drug concentration used versus the GABA concentration in the brain might preclude these drugs of potentiating GABA currents in the brain. I think that's what the question is also hinting at.
So the question is the drugs is the drug concentration.
In combination with GABA, also with GABA concentration in the synapse. Because GABA, very high GABA concentration might preclude the effects of these drugs in the synapse, or rather, do you think they might?
So the question is I just feel like the GABA concentration is kind of like high in the synapses, and
it's high, but it's also, I presume it's very transient, it's very short-lived, presumably. I don't know, I'm just assuming that it behaves similar to glutamate, but I might be wrong.
Yeah, it's kind of like I also don't have a very clear idea. I know GABA receptor, micromolar is not a very hard deal. It can also reach millimolar. But our results is I'm not sure. I just heard the question is the medicine will potentiate the drugs will potentiate GABA receptor, and this is caused from the high concentration of GABA. Is that question? I'm sorry. I just
No, I think it was just whether in the synapse these drugs would have an effect given the exposure to physiological GABA concentrations. But we don't know the concentrations of the drugs in the brain, presumably.
We know. Yeah.
We know? Okay.
Yeah. I'm sorry. I maybe I go too fast. I escaped that part. All the drugs we are using, because they're all clinical drugs. There's already concentration of how much. What we are using is pharmacological relevant. We are using it's relevant to the pharmacological concentration. So it's indeed real happen in the person.
Okay.
Yeah.
So then the next question is, sorry if you already mentioned this, But have you tried heterologously expressing the NL2 and or GAR LH4 subunits to see how they affect receptor function? So these the proteins that co-precipitated with GABA receptors, do they modulate activity of GABA receptor?
Yeah, we plan to do that, but yeah, we haven't done that part. But there's some great papers from, I think, from Yale, Susan's lab. They have investigated a lot of NR2 and GAR4, and they found the NR2 and GAR4, they didn't really modulate the GABA receptor channel function, but they will indeed regulate the clustering of the from the functional side. Yeah, but for the channel function, they do not show any activity on both of them. Yeah.
Both of these. Okay. So the next question, how was the yield of the protein? It must have been very challenging to obtain a cryo-EM grid feasible amount of native protein.
Thank you for the question. Kind of like, that's something I want to say. It's the yield is not that so bad. It's not that bad. It's kind of like really surprised. From 20 grams of brain tissue, finally we can get the protein concentration could be pushed to 6 milligram per ml. Yeah, but it's just maybe around 30 microliter. The concentration of final protein, the yield is not that bad. Yeah, for the cryo-EM, it's first when we figure out how to make it work, it's pretty challenging. But finally, it turns out to work pretty well. Yeah, and also that's the reason why we can see so many different compositions, because we have a pretty high concentration of the protein. So yeah, we can see so many compositions of GABA receptor. Yeah.
So just while we're talking about the purification, the aggregate peak that you see after, I don't remember whether it was affinity or size exclusion column, size exclusion probably, does that contain a lot of GABA receptors?
It contains some. Yeah, GABA receptor kind of like aggregate. It's not very, very stable. That's another challenging part. When no matter you are doing native or recombinant, you will match. Yeah, it's pretty easy to aggregate. It indeed contains some of the part, but it's also some like contaminated protein there from burning. They will form an aggregate peak there.
Do you have any idea what's in the aggregate? As in, could that bias the subunit composition that you're seeing because some tetramers are aggregating more than the others, for example, or is it just random?
It's pretty how to say, it will indeed affect your yield because some of the receptor is aggregate. But we indeed do mass spec to investigate what's made inside. But the subunits inside is kind of similar like what we get. So I kind of like how the composition could be in the similar way.
Okay, so the next question is, were you able to resolve any glycosylation? And if so, was its structure similar for each subunit across different subunit combinations? This might tell you something about where in the cell they were. Good point, actually.
Oh, the glycosylation. Wow. We indeed can tell the glycosylation. That's but for the glycosylation, for the people who don't know GABA receptor, so we mainly see alpha, beta, gamma. So they have different size of N-glycosylation and a different number of N-glycans. So that's the first way how we determine alpha, beta, and gamma. But I'm not totally sure how to base on the N-glycosylation to tell where it's coming from. So like which cell type, I didn't have that kind of like part of information. Yeah, but based on our resolution, we can indeed tell the glycosylation. Yeah.
And the glycosylation pattern, for example, for beta-2 subunit is always the same, no matter in which heteromer it is, or does it change depending on the heteromer?
No, it doesn't change. They all look like pretty similar. Yeah, pretty similar way. But some of them, they will like changing the number. Some positions will contain 3 glycans, but some positions will contain 2 glycans. But I don't know if that matters together with the cell type. Yeah, I don't have that part of the information. Yeah, but I'm happy, yeah, if you, yeah, can, like, yeah, I'm happy to know that part, yeah.
Okay. I was just wondering if I may, one more question. I think at the moment, we've been through everything in the chat. If I can bother you with another one. So I was surprised to see mystery densities. I mean, it's very exciting, but I was wondering, how did they survive the purification process? I mean, I know the tissue is frozen, flash frozen, but then the whole homogenization, wouldn't the ligand be expected to dissociate?
Oh, that's a super great question. Yeah, that's the question keep bothering us. That we will ask the same thing. Okay, if they are true, but what we see is that very strong. I can give you some example. Yeah, so recently published the rodent GABAA receptor. What they determined by the just native purification, they get the native the neurosteroid. Even after several wash, nanodisk, and several steps of purification, they still get it. Yes, that's one example. Another example is from, they are both GABAA receptor. Another example is from a recombinant structure. They try to get the APO, like without any ligand binding of GABAA receptor, but they still see a density as a GABA bind position. So it seems like, yeah, after so many steps, there was a wash or like, yeah, purification, it still binds there. So that gave us some hope, some like, yeah, some idea like, okay, it may still binds there. Something is maybe, yeah, make it like stay in the pocket and then still there. Yeah.
Okay, so maybe you mentioned it, so sorry if you did, but are the mystery ligands heteromer specific? Or did you find them across different heteromers?
Okay. So
So
you mean,
for example, I don't know, if we take lacosamide, I'm not sure how to pronounce that. So was it only at one subunit combination, or did you find it at different subunit combinations? Or I guess it's specific for one interface, right, between the specific subunits.
And it falls specific interface, so it can cross different kind of composition, and maybe two interfaces, they all hold different kind of like density. But yeah, it's based on what kind of interfaces contribute from the specific subunits.
Okay.
Yeah.
Okay, good. That was wonderful. I think I asked you everything I wanted to. We definitely, at the moment, we have no questions in the chat, and we are coming very close to the end of our seminar. So thank you very much, Zhou. This was really interesting.
Okay, thank you so much.